Welcome everyone. Welcome to this in-person colloquium. Fresh change after a long time to have somebody see in human flesh and blood. So welcome to this colloquium. So today it's a great pleasure to have Akshit Goyal from MIT. So Akshit did his bachelor's from St. Stephen's College, honoring in physics. And then he switched gears and joined NCBS to do biophysics, biological physics, or the physics of living systems. So he did his PhD there and then moved to MIT, where he is a Physics of Living Systems Fellow in the physics department there. And he works in a bunch of people, Alan Gore, Jeff Gore, and Sergei Maslow, and several people there. So he's broadly interested in how physics appears in the ecology of microbiomes. And he will tell us a broad overview of how to think about them today.
Can everyone hear me all right in the back of the room and on Zoom?
Just so people in here in person, please use the mic when you ask a question so that people on Zoom can also hear you.
Okay, I'm presuming that everyone can hear me okay. Thank you, Vijay, so much for the nice introduction. Thank you everyone for inviting me. I'm excited to tell you about my work today on something I call the statistical ecology of microbiomes, and this will become clear as I talk more. I want to begin with a brief introduction. My name is Akshit. I'm a theoretical physicist, and I'm currently an independent physics of living systems fellow at MIT. I have a background in theoretical ecology, complex networks, and computational data analysis. And as Vijay said, I have a bachelor's in physics from Delhi University. Oh, it says the speaker's voice is muted. Can we amplify this sound?
No, I can hear perfectly on Zoom.
Okay, I will continue. I'll try to speak louder if I can. Right, so I was saying that I have a bachelor's in physics from Delhi University, and I have a PhD in NCBS where I work with Sandeep Krishna. And currently I collaborate with a bunch of people, in particular Jeff Gore, Sergei Maslow, Arvind Murugan, and Otto Cordero. And my research focuses on microbiomes, which are communities of microorganisms that are ubiquitous on our planet. Now, I think the really interesting thing about microbiomes is that they're prime examples of complex systems with emergent phenomena. And wherever you might have heard about microbiomes, is sort of a booming field, you must have heard of them because of the macroscopic phenomena that they perform. You know, just to give you a few examples, they are responsible for a large fraction of collective cycling of nutrients across the globe. They're responsible for fiber digestion in your guts, for instance, digesting your lunch today. And they're also involved in a whole bunch of wastewater treatment. The challenge really is to control these macroscopic phenomena, to make nutrient cycles robust, to make fiber digestion healthy, and to make wastewater treatment efficient. However, how to currently do that using the microscopic components of these systems is what is currently unclear. Now, the microscopic components of the system are something that biology is really good at giving us. There's lots of sequencing and so on and so forth, which has given us a sort of parts list of each of these systems. And so we know from doing this that these systems have many, many parts, you know, over 10 to the 10 cells per ml. And moreover, unlike classical StatMech, where we might think of these parts as being identical, here they're evidently not. Thousands of different species can coexist and engage with each other in a dense web of interactions. What we've learned from this kind of characterization is that it's seldom just one or two species that are responsible for these collective macroscopic phenomena. Rather, all the species are collectively responsible for them. And even though we have lots of data about the microscopic components of these systems, we still have trouble connecting them with these macroscopic phenomena. Now, why is that? There are many reasons, but I would argue that the chief reason is that we currently group microbiomes at a rather arbitrary level. In fact, that level happens to be the level of species following sort of decades of ecology and evolution with plants and animals, where sort of people did really well by describing what this species does with that. Now, problem with using this description is that the species unit is fundamentally ill-defined for microbes. The standard biological species concept, which is groups of organisms that are able to sort of interbreed with each other and form fertile offspring, that just doesn't work for microbes because they reproduce asexually. Just one organism can give rise to two, and so on and so forth. And so biologists and ecologists understand this, and they critique this extensively, but there's no known alternatives that exist. And it's this lack of alternatives that I'm going to address today. In the absence of these alternatives, this is what the state of the art of microbiome data looks like. So what you see here are multiple samples of a particular microbiome from roughly the same environment. You can think them as sort of different parts of your tongue or different parts of your stomach. And so each column here represents a different sample, a different community. And within each column, you might notice that there are different colors. These colors represent different what are called species, right? And so each of these, and so the y-axis represents the relative abundance, or the fraction of each of these species in a sample. And as soon as you see, as soon as you start focusing on a species from sample to sample, you might start to appreciate why this is all sort of a mess. You can see that there's no reproducibility of species from sample to sample, and it really looks quite complex. And remember that I told you that these are all just communities that come from the same environment. And so, yes.
Is that also a very large number?
Yeah, that is a good question. Typically, it's of the order of 100, in some cases, a few thousand. A lot of it depends on the, because this definition of species is arbitrary, some of it depends on how exactly you define it, but the number of hundreds to thousands is generally agreed upon.
But wouldn't you define a species by its genetic composition?
Yeah, so that's absolutely correct. So the current way that we think of species and microbes like bacteria is by looking at the similarity of their DNA.
Yes.
But we don't focus on the similarity of their entire DNA. We focus on one particular gene, and what this gene is rather arbitrary. And so, we say that everything that has this one gene that looks the same, they're all members of the same species. If it changes a bit, it becomes a different species. And so because this definition is sort of distinct from function, as I'm going to describe, that's why it sort of ends up being a bad description.
Thanks.
And so this is an excellent question, which segues into what it is that I propose the field should be trying to do. What I think we should be doing is, instead of looking at these arbitrary sort of species level descriptions that are based on genetic similarity, we should be coarse-graining these systems into functionally similar units. And so my argument is that if you coarse-grain this sort of rather messy looking system into functionally similar units, you might see a much more simpler, a much more universal picture of these microbiomes. For instance, in this example, 3 coarse-grain groups in blue, red, and yellow.
Sorry, one question.
Yes?
So the samples are taken from where? All these.
Yeah, so in this case, they're all samples that are taken from one person's tongue a few millimeters apart.
Okay.
Does that answer the question?
Yeah. Okay, thanks.
Okay, great. So I was saying that we might see these 3 different coarse-grained groups. And the argument is that instead of this species level description where everything looked like a mess, here there's hope for some unification and understanding, because now when you look at the microbiomes in terms of these coarse-grained groups, the levels of each group are roughly the same as you go from sample to sample. That's why you see them as sort of these bands. Moreover, my argument extends further. These coarse-grained groups can form the basis for better effective models of these systems. For instance, if this is a community that performs polymer digestion, which is quite a common thing that microbial communities do, then the blue might represent a group of organisms that help break down the polymer into smaller oligomers. The red might represent a group of organisms that break down these oligomers into smaller monomers still, and the yellow might then represent a group of scavengers that further eat these monomers. How to do this kind of coarse graining is currently not clear, in part because the functions that these particular microbes perform is completely unclear. Even for a single species, we need hundreds of thousands of measurements, and we just got done saying that there are hundreds of thousands of species in each system. So in today's talk, I'm going to ask this question, which is how do we coarse-grain microbiomes in a principled, in a rigorous way that's not arbitrary? And I'm going to show you perhaps the first step that my research is trying to take to address this question. What I'm going to show is that we're going to start from an unbiased view. We're going to start, we are going to forget about species. We're going to start from perhaps the highest resolution view of these microbiomes that we can get. And instead of looking at static snapshots that I was showing you in the previous slide, we're going to actually look at the dynamics of these microbiomes over long periods of time. And so I'm going to sketch over the next slide what the central idea of our method is going to be. That I'm going to expand over the rest of my talk. Remember that we want to infer functionally similar units from data. What does the data look like? The data will look like the dynamics of various members of a particular community over time. And these dynamics are going to be at a high level of resolution. What does that mean? It means that we're going to be able to resolve two individuals that differ in even a single mutation from each other. Right? That's what I mean by high resolution. And so once we have these high resolution dynamics, we can ask questions about functional similarity couched in terms of the dynamical similarity between different individuals. We can start asking about statistical patterns of correlations between different individuals and start to ask how we can cluster them. And then finally, we're going to build mathematical models of these particular communities at different levels of resolution and ask which of them fit best to the data that we have, right? And I'm going to show you three different results. The first thing that I'm going to show you is that even really closely related strains, those that belong to the same species, even in sort of today's lingo, they're very, very similar in their dynamics up until a distance of about 100 mutations away. And after a distance of about 100 mutations away, they can completely decouple in their dynamics. And just to give you a sense of what 100 mutations is, mutation rates in bacteria are really quite large. And so 100 mutations can accumulate in a matter of just months. Okay, the second thing that I'm going to show you is that when we look more broadly at the community at distant strains, so not just close strains now, at very distantly related strains, we can see them often being much more dynamically similar or correlated in their dynamics than even their closely related siblings, suggesting that we can use this as a basis to now start clustering and asking a question in terms of dynamically similar clusters or units. Sorry?
This is a bit difficult to understand. Genetically, they are very similar, but
correct.
Functionally different. Why?
Genetically, they're very similar, but functionally different. Why? I'm going to answer this over the rest of my talk. The short answer is that it doesn't take many mutations to distinguish you in terms of what you're actually doing in the community. And what most microbes do in a community is consume resources. And so these mutations that we see in nature often seem to be concentrated in genes that make you different at consuming these resources.
Thanks.
Yes.
Maybe you should complete this slide.
Sure. Okay. So, okay. And so the last thing that I'm going to show you, and this is sort of going to be the punchline of my talk, which connects both of these components, is that by means of some really simple mathematical models and by means of a rather intuitive geometric interpretation of these models, we can recapitulate both sets of dynamical patterns that I will show you. And they will teach us that if we want to recapitulate these dynamics, then we have to be modeling these communities at the level of strains rather than species, which is what is conventionally done in the field. Yes, question.
Yeah, I think I'm still not very clear about why you are doing this sort of coarse graining, keeping the functionality as the axis.
Right.
Because I think, so what is the end goal is to understand the dynamics of this whole system to see to model it, I gather, but to understand what are the main nodes in this modeling. Because just to be kind of make a random analogy, if I were to group ecosystem, a macroscopic ecosystem in terms of something like vegetarians and non-vegetarians and okay, predators, prey, then of course they are in some sense functionally playing a same role in the system, but they can be genetically very different, predators, prey of different kinds. But maybe that's useful way of thinking about it if you're looking at the population dynamics, let's say, of that system, but it may not be useful if you're looking at something else. I mean, this way of coarse graining it is perhaps particularly useful for a particular goal in mind. And what are you losing when you do that and so on? Just if you can say a few words.
Yeah, that's a really pertinent question. I think the big goal in terms of why we want to coarse grain these systems is to solve this structure function problem that I posed in the beginning. The structure function problem says that there is some functional readout or some functional output of these systems. Let's say it's the rate at which they, there's some nutrient processing going on in your gut. And that's what you want to be able to understand, explain, predict, and control. But you want to do that using the microscopic components, the structure of these systems. The problem currently is that because the way that we currently look at the structure of these systems is in terms of these rather arbitrary units, any particular species or any particular unit that we use does not correlate with any functional readout that we try to take of these systems. And so that is severely hindering this field from being able to make this connection. And so the hope really is that the reason that that doesn't work is that because species are not the right functional units of these systems. If I knew, for instance, what the producers and what the consumers are, then I could write down a model in terms of the production of something and the consumption of something. And then if I coarse grain the data at that level, then I'd be able to make a better prediction.
So in the end, it is about. So in the end, it is about how what is most relevant for, let's say, maximizing wastewater recycling, or
Exactly.
what are the main, what is the the key nodes or the key
Correct. Yeah. So what are the nodes in terms of which we should understand these systems as performing and giving rise to these different phenomena that we can actually measure?
Okay.
So, I mean, this is not just a question of relabeling, right? I mean, this is the right degree, that is the right, but probably you also want to understand why the way it works.
Absolutely.
I mean, how is it the way it works?
Correct. Correct. Yeah.
Maybe the stability of the system, maybe
Right.
performance.
Yeah, that's a great point. So it's, of course, the other reason, the other thing that a lot of people say in microbiomes is that if I take, microbiomes have fingerprints. That is, if I take a sequence of your particular poop, it'll look completely different from my particular poop. But in the end, our gut is performing the same function of digestion. So there is some level at which there is stability of these systems that we just can't see if we start to ask questions about the species. That is, if we understood what the coarse-grained units are, we might also be able to posit what are the underlying functions that are giving rise to those coarse-grained units, or the stability of these functions that we see around us. Yes.
Can you hear? Yeah. Okay. So I actually have two questions. So one is, will you later on get into a bit more detail about what do you mean by dynamics? Because you had a plot where you had abundance as a function of time, and you said that was a dynamical sort of quantity.
I'm going to describe the dynamics in the next slide.
And so something like abundance as a function of time will be correlated in some way to what you're calling functionality.
Yes, eventually.
Okay. And a few slides ago, you had this plot where you had the abundance as a color map for different samples. Now, if I sort of take a very naive view and I say that if I have a microbiome, I'll put 1. If I don't, I'll put 0. Then, of course, on an average, I'll get some uniformly colored thing. And then if my criteria for classification is very coarse, then it's not unusual that I get something which is very well correlated across samples. So is there any sort of arbitrariness in terms of how far down do I want to go in terms of how much information I want to keep?
Yeah, no, this is an excellent question, which I'll come back to later in the talk.
Sure.
But yes, that's an excellent question. Of course, if you group everything into one group, then you'll get complete flat. So we don't think that that's how these systems are actually working.
Okay.
Okay. All right. So let me move on. Okay. So like I said, that was a brief summary of what is going to happen in the bulk of this talk. The first thing that we need to do is that we need to get the data. And so let me just describe you how we get that data. We need to track the eco-evolutionary dynamics of a particular microbiome and get that at high resolution. And so for this, my experimental collaborator, Otto Cordero's group, went to the forest and sort of sampled 10 different pitcher plants. And these are sort of plants that have these pitchers that you might have seen, and they have fluid in these plants, fluid in these pitchers, which have microbial communities that help the plants digest and eat insects. Without these insects, these plants really cannot, without these microbial communities, these plants really can't eat the insects, right? And so that detail aside, is just a particular microbiome that they worked on. They can now sample fluid from these pitchers, isolate microbial communities, and start growing them in the lab because they want to have controlled conditions. So you start growing them in a medium that's not very different from the medium in their natural environment. What that medium contains is essentially a bunch of resources. There are resources and there are these microbes, which if you put, they will deplete the resources and grow. After some point, they've depleted all of the resources in their environment and they've grown. This takes a few days, after which you take a small fraction of them and transfer them to a fresh medium, right? And so over time, this process of growth and transfer continues. And you can track these communities at a rather coarse species level quite easily. What you do is you just sequence this one gene that I was talking about that distinguishes species from each other. And as a result, you get data that looks like this. On the y-axis of this data is the relative abundance. Every column is a particular community, and the x-axis is time measured in the number of transfers that the experimentalists are performing, right? And each different color here is again a different species. So there's a gray species, a pink species, a green species, and so on and so forth. And what we saw was that very quickly, these communities reach some sort of steady state. That is, if you start to look at the abundances of gray species, that doesn't change too much from transfer to transfer. If you look at the abundance of the pink species, it doesn't change too much. And so approximately for most species, that is the case. And so we can now just use this experimental platform. And what my collaborators did was to just continue this experiment for a whole year to get the long-term dynamics of this particular community. And again, what we saw here was that there were no dramatic changes in the abundance of any particular species. Yes.
What distinguishes white from blue from green, red? What's the parameter? What distinguishes the
gene? So what you do to get these data is that you essentially crush up all of the cells and you isolate this one gene that is common to all bacteria, and you sequence just this gene. Yeah, and for every individual that has the same, where this gene reads the exact same, you call it one species. That's the gray species. If there's even a one-letter change in the spelling of that particular gene, you call it a different species, and so that's a different color, and so on and so forth. In particular, there can be many changes that distinguish a species from the other, but that's the bare minimum. Okay, great. So.
Sorry, but I thought it's supposed to be 100 or so, or 1,000.
Correct. Yeah. So that is an excellent question. So it's 100 or 1,000 in this particular picture in the natural environment itself. A peculiar thing that happens not just with this experiment, but with most microbiome experiments is that when you bring these rather complex communities to the lab, a large fraction of species die. And so what you're left with is something of the order of 20 or 30 species instead of a few hundred. This is a common thing that happens. That is sort of the cost you pay for controlled data. Right.
I have a question.
Yes.
So I guess in the pitcher plant, it's growing. It's not a well-mixed condition. Whereas in the lab, in the abundances that you're measuring are out of a well-mixed test tube. right? So if you were to do this, let's say from a colony rather than from a tube, how would these abundances be then?
Okay, yeah, that is a good point. I don't know to what extent we can say that the picture is not well mixed, because maybe the picture is not super representative, but these aren't really tubes. These are really like conical objects that are much like, a small water bottle. And so, I mean, we don't know to what extent there is spatial structure in these communities. I mean, there must be to some extent, but some people have done studies to sequence stuff from the edge of the plant and so from the picture in sort of different areas. And what they found is there's not very large spatial structure that's going on in these pictures. Does that answer your question, Shashi?
Yes and no. I mean, so I guess, first of all, when you grow it as a colony versus in a tube, these abundances are going to be vastly different just because of the spatial nature of growth and so on, right? So I guess in the picture, I find it hard to imagine at least that it's perfectly mixed. There probably is a combination of both spatial and some well-mixed growth.
Yeah, it's definitely not perfectly mixed in nature, but my sense is that since my collaborators sampled from the bulk and not, if there's some spatial structure, it'll probably be on the walls or at the floor of the picture. And so, yeah, I mean, we can talk more about this, but essentially the idea is that we don't think that spatial structure is going to be a huge problem here, but certainly it could be.
We can catch up.
Sure. Okay. Any more questions? Okay, I will.
Sorry,
yes.
just one. You had 21 travelers, 46. Those are the number of species, or no?
What's this is time measured in terms of the number of transfers. And just to remind you,
that's transfer. Yeah, I understood what the transfer was. Yeah. Oh, that was transfer, not traveler.
Yes. Sorry about that.
No. On the top you had 41 transfers.
Transfers.
Okay, that was just a general question there. When you're doing this in the lab and you do this transfer process, could you be producing mutations that you wouldn't produce in the actual picture because the conditions are now maybe much more enriched or whatever? They're definitely very different in this transfer.
Yeah, no, that is an excellent question. And in fact, to be honest with you, the reason that my collaborators actually started doing this experiment was that they were actually interested in what kinds of evolutionary processes they'll be able to observe in terms of what kind of mutations will occur and how will species co-evolve with each other. What we found was that there's not really a lot of that went on in these communities. What happened largely was ecological dynamics. And so even though we call these eco-evolutionary dynamics just because they occur for a long period of time, for many, many hundreds of generations, we didn't see a whole lot of mutations, even though you're right, we certainly could. Okay, so like I said, we get these long-term ecological dynamics. And what we see is that most species are sort of, there's some sort of drift around an equilibrium, which is to say that there's some sort of fluctuations around steady state that each of these species is showing. And just to show you sort of a zoomed in view of just two of these species, here are two species again in this low species resolution. There's a blue species and there's a pink species. The names are not important. And so this is roughly the species level description is, like I said, where everyone would stop. But remember, we wanted a high resolution view of these communities. And so what my collaborators did was that at 8 specific time points here, they performed what is called deep sequencing, where instead of sequencing this one gene that I was sort of talking about, you kind of crush up the cells and sequence everything that you can. And then it's a job of sophisticated computational algorithms that you can write to be able to sort of reconstruct and backtrace each of the small reads that you get to be able to get the dynamics in terms of this community. This is something that I did that I'll be happy to talk about the details of offline. But suffice to say for now, what you get as a result of this are the dynamics of these same communities at a high resolution. And so just to show you what that looks like, things can often look very different. For instance, we learned that this blue species that I was showing you on the left is actually composed of 2 different strains. one strain 2, which I'm showing in blue, and one strain 2, which one strain 1, which I'm showing in blue, and one strain 2, which I'm showing in green. And what we learn is that the strain that strain 2 is relatively static in its dynamic, in its sort of abundances over time, whereas it's actually strain 1 which is responsible for most of the dynamical fluctuations of the species. In another species, things can look different. Here again, we have 2 different strains that this pink species is composed of, but here both of the underlying strains are quite dynamic. And in a third species yet, which I haven't shown you here, we see that the species abundances are quite static, and it's the underlying strains that can be quite dynamic, right? Yes.
Your experimental friends keep on changing the the nutrient content, just experimentally day after day. Do you think you'll see a difference in the way these plots are? Actually, the dynamics will change.
Yeah, so just to confirm what you mean by changing nutrient.
Yes, I mean the environment in which the bacteria are living, actually. So first you took it out of some plant.
Right.
Now suppose, but there are many other things in the world, actually, like my gut.
Correct. Oh, yes,
and you
great.
change it, then is there a difference?
Absolutely. I think there absolutely will be. In fact, there's not even a guarantee that these species will be able to survive in that environment at all. And in fact, one reason why this experiment was constructed to mimic the conditions of the picture as much as possible was that that's the only way you can get a high diversity to exist in the controlled conditions of the lab. So certainly that would change things, but I think in a way that would be actually very difficult for experimentalists to study, just because everything will die.
Okay, thank you.
Okay, so
I had a couple of questions. So you identified species by just looking at a particular gene, right?
That's correct. That's
so
correct.
now, supposing there are 2 members with, but now you again look at you do this deep sequencing where you look at all the genes.
That's correct.
So now it's possible that even though the gene by which you identified them are the same, 2 guys might differ in some other genes, right?
That's perfect. That's exactly how these 2 strains are the same, are belonging to the same species. If you look at that one gene, they look the same, but it's in the rest of their genome that they look different. Correct.
Okay, and how big is how many genes are there in all?
Yeah, so there are of the order of 1,000, in fact, about 5,000 or so genes. And so, these genomes have about 1 million different base pairs, or 5 million different base pairs.
Okay, so depending on which gene you look at, their evolution might be quite different. That's what you're saying.
Absolutely.
Right? Okay, thanks.
Okay, I will move on. So like I said, this was just a zoomed in view to show you how we get these dynamics and that we can get them. But remember that we have not just the dynamics for these 2 species, we have them for all of the species in up to 10 different plants. And so now with these hundreds of different strain pairs, we can start to ask questions at a statistical level. That is, we can do statistical ecology. And now we can get back to this question that I posed at the beginning of my talk. Remember, what we want to do is that we want to be able to coarse-grain strains that are functionally similar. We still don't have an idea of what functions these strains are performing, but we do have an idea of how they're dynamically behaving in the context of this diverse community. And so we had the following hypothesis or idea, which is that if 2 strains are functionally similar, then they ought to show very similar dynamics in the context of the communities in which they reside. And this is the idea that we're going to use for the rest of our statistical analysis. And so with this, we ask our first question, that is, even within the same species. Yes. Yeah.
So since you are, so since you are ascribing some importance to the fluctuations, I mean, because as you said, a large fraction of your species die, how relevant would be the statistical study of these fluctuations in these controlled settings compared to the sort of real life settings that you flashed towards the beginning of your?
Yeah, no, that's an excellent point. So the species that die, they die within the first 1 or 2 transfers. And so you won't even really see them in these dynamics. it's certainly true that the nutrient environment that is collectively created by hundreds of species might be different from the nutrient environment created by these dozens of species. And so you're absolutely right that we are making the assumption that we can learn what we learn sufficiently from these dynamics maps to those conditions. We don't know if that's necessarily true, but at least we can start somewhere and see. The hope will be, as you'll see at the end of my talk, that we can now take this approach to real ecosystems without bringing them to the lab and be able to ask the same questions.
Thanks.
Okay. All right. So like I said, we can now begin to ask some questions about the statistics of these dynamics. The first question that we're going to ask is, within the same species, do strains within the same species show similar dynamics or different dynamics? And just to show you a cartoon of what I mean, say that we have a community with different species in different colors. Let's say I focus on this particular species. I might see two of its strains. Let's say I call them the major strain and the minor strain, and these are their dynamics over time. I might see that these strains are coupled. That is, they're very similar in their dynamics. They have highly correlated dynamics. Yeah, no, that's absolutely correct. So by strain, I mean basically anything that is within a species. So if a species is composed of 3 genetically distinct sets of subpopulations, each subpopulation is what I will call a strain. Okay, great. Right, so within each species, we find a pair of strains, and so those pair of strains might be highly correlated in their dynamics. That's when I'll call them being coupled. Or if I zoom in on another species, let's say this purple species here, I might observe its constituent strains, the major and the minor strain, to be completely decorrelated in their dynamics. And then that's what I'm going to call decoupled strains. And so motivated by this idea, we're going to measure the temporal correlation of the trajectories of strains of the same species. And we're going to take a statistical view. So I'm going to draw a histogram here. The histogram is going to be this quantity I call the strain-strain coupling. That's nothing but the magnitude of the correlation between strains of the same species. And just to orient ourselves on this plot, on the left-hand side of this plot, we have low coupling that's going to represent decoupled strain pairs. And on the right-hand side, we have highly coupled we have highly coupled strains. And here's what the data look like. What we see is a bimodal distribution with one peak composed of highly coupled strains and the other peak composed of highly decoupled strains, separated rather cleanly from each other. Yeah, so what I do is that I have 2 trajectories, one for each of the strains, and I can literally just measure the Pearson correlation between them. And that the strength of that correlation is what I'm plotting on the x-axis. Okay, so we have coupled and decoupled strains, and we really want to be able to understand which strains ought to be coupled and which strains ought to be decoupled, so that we can use that to be able to group coupled strains together and keep apart decoupled strains from each other. And so that's the next question that we're going to ask: what determines which strains are coupled versus decoupled? And here yes.
I have a question. So if you did this for, say, arbitrarily 2 different species, you would have a peak at decouple. Is that what you would expect?
If I did this for different species, I will in fact do this for different species in a few slides. Okay. So maybe you'll get your answer.
Okay. Sure.
Okay, great. So we want to ask whether there is a good predictor for which strains of the same species decouple or not. And so here sort of our hypothesis was that the genetic distance, not over this one gene that distinguishes species, but over their entire genome, might be a good predictor for which strains couple or decouple. And so I'm going to show you that here by means of this plot. On the y-axis, I'm going to plot the strain-strain coupling. That's what was on the x-axis before. This is just the magnitude of the correlation between strains of the same species. And the x-axis is going to be the genetic distance between those strains. And this is measured in the number of mutations or the number of SNPs distinguishing them. And note that this number can range anywhere from 1 mutation distinguishing 2 strains to up to 10,000 or so, right? And so this is what the data look like. Each point here is a pair of strains that belong to the same species. But I want you to focus on this dark red line, which represents the moving average. What you see in the moving average is that if two strains of the same species are a very low distance from each other, they are usually coupled in their dynamics. Their dynamics are highly correlated, but there's a sharp transition at around 100 or so base pairs where these typically become decoupled, right? And so we can use this idea to sort of draw this rule of thumb at about 100 base pairs, where we can say that 100 mutations might separate dynamically similar from dynamically dissimilar strains, right? And so now at least we have one criterion to distinguish strains that belong to the same species. What we're going to do next is to ask this question at the level of the whole community, that is strains that belong to different species.
Sorry, I'm just a little confused. When you say 100 mutations is 0.01% of the genome, then 10,000 is essentially the whole genome.
10,000, sorry, 100 mutations.
Is a 1%,
Yeah.
0.01% of the genome.
That's
it's
correct.
1% of the genome.
Correct. So
is
it's about 1 million or so mutations that you need to
to
distinguish.
get a different species. Okay,
Correct.
so that's what.
And this is in bacteria. So bacteria about 1 million base pairs, but in humans you need about 1 billion.
Okay.
Right. Okay, so we did stuff within strains of the same species. We looked at very close scales. Now we're going to look at sort of community-wide scales. And here the question that we're going to ask is one about interspecies interactions. And usually, again, the convention in this field is that these interactions occur at the level of species, right? Yes, question?
If you go back to the previous slide.
Yeah.
So the decoupling transition that happens around 100 SNPs, right?
Yeah.
Is that also a statement about how functionally different things come up?
Absolutely. And you will see as I come to modeling these dynamics, we are going to be able to understand how a functional distance between strains leads to this decoupling.
Okay, so the things that are coupled are functionally the same.
That's correct. Okay, so like I was saying, usually the convention in the field of ecology is that the interactions in a community occur between species, right? But again, remember that this doesn't need to be true. We just learned that strains that even belong to the same species can be very, very different. And so since we have these data, we can now start to ask, are the interactions that are in a community stronger at the level of species or at the level of strains? And so the way we're going to do this is sort of motivated by the following fact, which is that that this experiment was done in a relatively stable environment. The nutrient environment was well controlled. And we have a large number of species that are sort of all fluctuating around a steady state. And so in these conditions, if there are interactions between members of a community, then we should see them popping up as correlations between these members in terms of their dynamics. That is, if there were two strains that were positively interacting with each other, then if the abundance of one goes up, we should see the abundance of the other go up as well. And so sort of motivated by this, we're going to ask about the community-wide correlations at two levels, at the level of species, which is going to be on the x-axis, and at the level of strains, which is going to be on the y-axis. And let me describe what I'm going to do on each of these axes. On the x-axis, I'm going to measure what I call the species-species correlation. So I'm going to take any two pair of species, call them A and B, in my entire community, and I'm going to measure the correlation between their temporal trajectories, right? And that's what I'm going to plot on the x-axis. There's going to be 1 point for every pair of species. And for those same species A and B, on the y-axis, I'm going to zoom into each of their strains. So species A might be composed of strains A1 and A2. Species B might be composed of strains B1 and B2. And we already looked at the sort of correlations between strains of the same species. So we're not going to look at that. We're going to look at correlations between strains of different species across these boundaries. And there are 4 different correlations that I can measure here. I can measure A1B1, I can measure A1B2, A2B1, and A2B2, right? And what I'm going to plot, since I need to plot 1 point here, is that I'm going to plot the largest of these 4 pairwise strain correlations for that particular species pair. The argument being that if any one pair of strains from across a species boundary is more correlated with each other, then we want to know that, right? And so just the argument is going to be every point is going to be a pair of species. And if that point lies in this orange region above the x equals y line, then we're going to say that for that pair, the strain correlations are stronger. And if it lies in the white region, the species correlations are stronger. And here's what the data look like. Remember, each point here is a pair of species, right? And what we see here is that the majority of points, 76% of them, in fact, have strain correlations being higher than species correlations, suggesting that the interactions in this community are presumably stronger at the level of strains rather than species, with a huge caveat that correlations are not in interactions. I apologize for that little bit getting cut off. But the caveat is that correlations are not in interactions, and we're going to get to that. But before I get to it, I want to sort of situate you on this plot and show you two interesting behaviors that emerge from comparing these trends of correlations. And there are sort of two regions that I want you to focus on. The first is this region on the top left, which is a region that which contains what I call surprises. These are essentially regions where if we focus on any one species, let's say any one pair of species, let's say this blue species and this red species, at the species level, we'll see almost no correlation between their dynamics, which might lead us to assume that there are no interactions between them. But if we focus on each of their minor strains, for instance, that is, let's say if I focus on the minor strain of the blue species, that's in blue on the bottom panel, and the minor strain of the red species, which is in red on the bottom panel, what sort of emerges is a nearly negative perfect correlation between them. And this sort of correlation or presumed interaction, I would argue, could have only emerged if you were looking at these communities at the level of strains and not at the level of species. The second sort of interesting region of this plot is sort of here on the bottom right, which contains what I call exceptions to sort of what we usually see. And here, an example might be this sort of blue species and this red species, which are relatively highly correlated in their dynamics, leading us to think that they're interacting. But if we focus on any one species, let's say we focus on this blue species here and looked within it for its strains, what we see is that there's this blue shaded region, which is one strain, and this green shaded region, which is the other strain. These two strains are essentially decoupled from each other. These strains are sort of doing their own thing, which might not surprise us then why, in this case, the species correlations were larger than the strain correlations.
So sorry, so the right-hand side, you're saying it's essentially like a coincidence that they were correlated when you looked at it on the species level?
Yeah, that basically essentially that is correct. Yeah, that we saw some essentially
correlation, but they're actually uncorrelated at the
correct.
If you
Or either that, I can't distinguish it from this, what you just said, and the second possibility, which is that the species themselves are correlated and interacting, but the strains within them have a high degree of sort of neutral fluctuations that are occurring and leading to sort of essentially decoupled dynamics. And so that's actually something important that we're going to come to in the next slide.
And on the top left corner, the ones you showed, yeah. The actual number over there is very small, probably, right? Or as a fraction of the total number of points on the graph. Roughly what fraction is
about 6 or 7% fault points
of the say? Okay. And so there, when there's a correlation between the strains across the species, that didn't contribute to the species correlation because that was sort of
they were the minor strains, and so
the minor strains cancelled it out in some way.
Essentially the correlations were sort of dominated by the major strains. That is, the strains that were in much higher proportion. And so, that's why the correlations are masked at the species level.
So when you compute the species, you weighted with the abundance of the different strains. So it's not just an equal weightage. Okay, thanks.
So yeah, I mean, actually, it's a related question. Yeah, it's a question related to that question. So because I can think of the species correlation as some form of an average correlation. I mean, it should be like this, right? That because the y-axis is choosing the maximum out of. So the y-axis is choosing the maximum out of them.
Yeah.
So in that sense, I mean, if you had done some other sort of averaging over the strain correlations, would this feature have gone away that much of the points are on the upper left or corner of the...
Yeah, no, that is an excellent question. And in fact, there's a whole battery of tests that we put this particular result to. Some of those tests I'm going to show you, but then some others I'm not. So let me assure you that here. You're absolutely right that on the y-axis, I'm comparing the maximum of 4 numbers, but on the x-axis, I'm only looking at 1. So maybe it's no surprise that I see most of the points above the x equals y line. And they're sort of, so the question that it leads to is one of what your null expectation should have been, right? Like we see 76% of cases strains being higher. What should it have been if there were no correlations, right? And so there are many things that we can do to address this. One that I'm going to show you. The other is sort of, you can take the same data, you can sort of shuffle all of the species and strain labels around, do the same calculation, which is subject to the same artifact of comparing 4 versus 1. And then what you start to see is indeed, you see a bias. That is, it's not 50-50, but the bias is much lower. In fact, it's sort of the order of 55-45.
I see.
And so anything above that 55-45 level, which is certainly above the level of statistical significance that we observe, is grounds for thinking of this as a real signal.
Okay, and so if you sit at a particular species correlation on that plot, can you tell me, like, what does the distribution of the strain correlation look like?
That is, you mean you're asking for some sort of conditional? That is,
yeah,
conditional
so suppose I condition on a particular species correlation. So what's the distribution of the strain correlation given a particular species correlation?
That's an excellent question. That would just be the margin of this distribution, right?
Right.
I don't know off the top of my head what that marginal is like, but I'm happy to look at it for you later.
Sure.
Okay. So there's a text. Yes. Go ahead.
Okay, so I have a nice question that across the species, if you look at different major and minor strains, is the relative abundance roughly the same across the species? Like all the major strains occur maybe 70% times or something? Or does this weight also varies across the species?
I see. Let me see if I understand your question correctly. So are you asking within each species, your major strain could be at some high relative abundance and your minor strain could be at some low relative abundance? Is there a typical relative abundance?
Yes.
Okay.
So is roughly that constant across the species? Because otherwise this graph would are weighted with these WIs and WJs, which can mask a lot of things and puts, even if there are like large strain correlations, because of this weighting factor, it can make it across 45-degree line or the other way around.
Yeah, no, that's a good question. So typically, of course, there's a broad spread here, but typically, your major strains are anywhere between 70 and 90%, and your minor strains are, of course, 100 minus that. But of course, there's some width to this distribution that then I'm not sure whether it is consistent with your masking point or not. But
I see. Thanks.
But yeah, I'll be happy to talk about this more. Okay, there's another question on the chat, which is, what if correlations are site specific within the genes that are being sampled? I see. So just to see if I understand this question correctly, site specific, as in what is the correlations?
Can I try speaking this question out?
Yes.
Am I
yes,
audible?
Please. Yes, please. Yes.
Yeah. What I meant is you seem to be comparing two different sequences, whether of DNA or of RNA. It isn't clear to me. Nonetheless, if the variation in any sequence is highly restricted to one region or two regions, the bulk of the comparisons would show very little correlation, and the one that actually is being strongly selected for would be masked out.
Right. Yeah. Right. So just to clarify, we're not looking at the correlations between two DNA sequences. We're looking at correlations between the abundance trajectories, that is the abundance over time curves. And so even though we know that these abundance over time curves belong to strains with a certain DNA sequence, I don't think that they will be particularly masked by the sequence differences between the sites. Did that clarify?
Maybe I can bring it up at the end later. Please carry on.
Sure. Please do. There's another question.
Yeah, this question stems from my not knowing how the strain correlations are actually calculated. So you showed that these are the temporal correlation between the relative abundance, right?
Yeah.
So if the way you showed it for coupled strains, you sort of have similar trajectories. But now say if it might be completely wrong, but if one of the strain is actually say transforming into another, there would be some sort of a negative, like one of the abundance would decrease while the other increases. Will that also show up in the strain correlation being high, the way it's calculated?
It definitely could show up. Yeah. If one strain is replacing the other,
yeah,
that's something that could show up, but it won't show up in this plot. In this plot, the y-axis is being measured for strains that belong to different species. So we never measure strains that are replacing each other.
Right. Sorry. Yeah.
Okay. All right. So we looked at two regions of this plot. Now we go back to this question of what does this pattern of dynamical correlations tell us about the interactions that are occurring in this community? And in particular, this is a question about, what should our naive expectation of this pattern have been in the first place, right? And so here we can sort of ask the question by using theory to guide us and ask what null model of the ecosystem and its dynamics would give us this kind of pattern and which wouldn't it? And really there were sort of two broad hypotheses that were in our mind. The first was one where strains are actually functionally distinct from each other. That is, strains are sort of the unit of interaction in this community. How different in function? Well, one thing that we noticed was that most of the mutations that were occurring and that were distinguishing strains from each other were mutations that were concentrated in metabolic genes. And this maybe speaks to your question, Vidyanand, which is that basically most of the differences between strains had to do with how these strains use different food sources. This is not very uncommon in these microbial communities. And so this suggests also the kind of model that we should be building. The second is a contrasting hypothesis where we argue that strains are essentially functionally identical, but it's actually a whole bunch of random fluctuations that occur in these communities, including a lot of the statistical artifacts of the kind that were mentioned in a few of the questions that are leading to this kind of pattern, right? And so we're going to use models to be able to distinguish which of these hypotheses is consistent with the data. And what kind of models are we going to build? We're going to build what is called a consumer resource model in ecology. And this is a broad family of theoretical models, only one of which I'm going to show you today. There's a whole bunch that I'm going to show you tomorrow, for those of you who will be here tomorrow. But here's sort of the broad idea of what a consumer resource model is at its heart. As the name suggests, the model has two kinds of ingredients. There are resources that are sort of present in the environment. There's sort of different colors or different resources in this cartoon. And there are different consumers of these resources. There are different species, and for our case, strains within these species, right? And these strains consume these resources at certain rates, and they use, and by consuming these resources, they get energy and they grow. And sort of the dynamics of this model are sort of the simplest feedback loop of this kind that you could imagine. The dynamics simply say, if any strain has a surplus of resources, a surplus after paying a certain maintenance cost, then it can grow. And as a particular strain grows, it depletes the resources that it consumes. And that sort of results in two sets of coupled differential equations. The first set is an equation that's sort of symbolized by N, which is one equation for each strain, with the subscript I. And the second is a set of equations for each of the resources, which are symbolized by R and the subscript alpha. And each of the strain equations just say that there's a maintenance cost MI that each strain needs to be able to pay. If it doesn't pay it, then it dies exponentially at this rate. And by consuming resources, the more and more resources you consume, the more and more energy you get by consuming these resources. And so if you consume energy and get more than your maintenance, then you grow exponentially. And as you're consuming these resources, the resources get depleted until they're no more. Right. So the size of the CI alpha, which I'm going to show you in more detail, is there are rows which all represent the number of strains in your community, and there are columns which all represent the number of resources in your environment. And so that's going to look, in this particular case, roughly as a 30 by 30 matrix. How do you find the entries of this matrix? Yeah, so that's a good question. We're going to look at random versions of these matrices, and it turns out we're going to be able to learn a lot from that. We don't have any hope of finding the right parameters from the kind of data that we have. And so that's why we're going to ask questions in terms of the generic features that we can see in this model. Right, and so since this was already, there's another question, which is, aren't resources constantly replenished? Okay, that's an excellent question. The resources will be replenished, not constantly, but in periodic intervals of time that have to do with, we're going to mimic our experiment. So every transfer, we're going to supplement a bunch of resources, and they're going to be depleted. I'm going to show you exactly what we do soon. Right, so I was saying, so these are the equations, and really it's the matrix C, which describes the functional structure of the strains of the species, right? And what does this matrix C look like? It looks different for each of our two hypotheses, but broadly, it looks like a matrix where all the rows are our different strains. So, here in this case, it might be a strain A1, A2, B1, B2, and all the columns are different resources. And so just for this cartoon, I've shown you 4 different resources, R1 through R4, but in principle, this could be as high dimensional as you want it to be, as long as you can do the simulations. And the idea is going to be that in the first hypothesis, strains are actually distinct from each other. There is strains A1 and strain A2 are different. And so we're going to have a parameter D, which is going to be the only real parameter in our model, which is going to be a tunable distance between strains of the same species. If this distance becomes 0, we're going to say those strains are completely identical in how they consume resources. And if as this distance D keeps increasing, strains are going to become more and more different. Yes? Okay, that's a good question. So the CI alpha represents sort of the growth rate of the sort of instantaneous per capita per unit concentration growth rate of each of these strains on each of the resources, right? In this particular model, I've assumed that it doesn't change with time. There are others that have brought up this idea that it's actually not dependent on time strictly, it's dependent on the concentration of the resources. That is, when the resources are very, very large in abundance, there's some saturating growth rate that you reach. And as these resources get depleted lower and lower down, this sort of rate goes down. So, for simplicity, the model is built so that we imagine that we're in this linear regime. But in principle, the the qualitative behavior of the simulations that I'm going to show you doesn't change by assuming sort of different forms of the CI alpha.
What is the distance d? Can you explain that?
Yes, yeah, okay. This is a good point, and I was going to get into it, which is that the distance d between two strains tells you roughly the mean square difference in the growth rates of any two strains on any particular resource on average.
Will that be mapped to the number of mutations?
We will in fact map it to the number of
Okay, so that's the
mutations.
distance, the decoupling.
That's correct, and yeah. So right. So here in the model, the distance d really is an average difference d between how strains are consuming resources. It's a functional distance, and so that's something that we're going to control. What about the distance between strains of two different species, strains A1 and A2? We're going to assume that there are two random vectors in this resource space. And I'm going to describe what I mean by that by means of a geometric interpretation in a bit. But for now, we're just going to initialize sort of two random vectors, one for A1 and one for B1. And so they're going to be some random distance away, right? So that's hypothesis one. Hypothesis two, remember, was one where species are really the unit of interaction. Strains of the same species are identical to each other. And so that's just going to correspond to a case of this model where D is 0. So now strains A1 and A2 look like the same. What's going to happen in this particular model is that the reason why you get that the two strains within the same species are not the exact same is that there are stochastic sort of birth-death noise or sort of various stochastic fluctuations that are occurring within the species. And so we're going to do that in the model by means of a sort of root n noise that we can add to these equations. Right? The details of how exactly these simulations are done are not so important, but I'm happy to talk to you about them after. What we now can essentially do is that we can simulate the kinds of growth death cycles that we do in our experiments almost exactly, where again, just to remind you, we sort of initialize the medium with a whole bunch of nutrients or a whole bunch of resources and a small population of each of our strains. They then grow by depleting all of the nutrients. Once they've depleted all of the nutrients, we take a small fraction of them, transfer them to a fresh medium with just freshly replenished resources. I hope this answers the question that I was asking, that you were asking, Nandanaya. And then we just keep going on and on. So we simulate the dynamics of these serial transfers exactly like we did in the experiment. What generates the sort of fluctuations that we saw in the experiments is the fact that this medium is actually quite complex. It contains tens of different resources. And just like the experimentalists can't perfectly capture the exact same nutrient medium composition from one transfer to the next, we're going to assume that the growth medium between two different transfers is slightly different. That is M and N and M prime, just to conceptualize it. And now we can run simulations and we get a whole bunch of dynamics. And it turns out that this model is actually really good at being able to recapitulate the dynamical patterns that we saw in the experiment. The first pattern that it captures is that it recapitulates the distance-dependent decoupling that we saw between strains. And this is how we're going to see it. The y-axis, just to remind you, is what I call the strain-strain coupling. That is correlation between strains of the same species. And the x-axis is this functional distance d that we could control in our model between two strains of the same species, right? And this distance is in units which are normalized to the average distance between any two species. And so a distance d of 1 will imply that two strains are about as different in consuming resources as two random species. A distance of 0.1 will imply that they're about 10% as different as two species, and so on and so forth. And here's what the results from our numerical simulation of this model look like. Again, each point here is a pair of strains from the same species. But again, if you notice the dark red moving average, we observe that when this functional distance d between strains is, of course, very small, the strains are essentially perfectly coupled because they're essentially the same in how they consume resources, but that there's a transition beyond d of about 0.1 or so, where the strain-strain coupling can be quite low. And this is quite reminiscent of the data that we got from our communities, where instead of the functional distance d between strains on the x-axis, we have the genetic distance d, the genetic distance between strains measured in the number of mutations, right? And there's one important thing that the model is trying to teach us that I want you to keep in mind for the geometric interpretation that's going to come in a couple of slides, which is the fact that the distance between strains can actually serve as a proxy for the correlation strength between them. The lower the distance, the higher the correlation strength. And that's something that we're going to use later on. The second pattern, yes.
Okay, this distance is not at the genetic level, right? The distance d in the model.
So yeah, there's no notion of genetics in the model.
Right.
There's only the consumption coefficients of each of the resources.
So it's more of a functional distance rather than
Absolutely.
a genetic distance.
Absolutely.
So why should that correlate with the genetic distance plot?
So the real answer is I don't know, but I can speculate, and my speculation would be that the mutations that we see in these strains are not random, but in fact are concentrated so that in certain sort of resource consuming genes in a way that you sort of, every mutation that you get sort of makes you a little bit different in how you consume resources. One sort of easy way to think about this is that a lot of these genes are involved in the transport of nutrients from the outside of the cell to the inside of the cell. And so a lot of the mutations can change the affinity of these transporters to particular nutrients. Yeah.
You mentioned about, when I asked this question about C alpha I, the matrix, you said that, of course, it's not, determined experimentally, but there's some random matrix or something.
Correct.
So now the question is that you didn't explain what you really did to get these models, graphs, right?
Okay.
What did you do? I am very curious to know that, actually.
Sure. Right. So, sorry, I can explain. Basically, what you do is you do a whole bunch of different simulations where you create different communities. You can sort of imagine one community that is representative of each plant that we did the experiment on. And each sort of community is composed of a pool of strains that is different from community to community. And so the way that we do that is that for each particular community, I instantiate, sorry about that. I instantiate one random version of this matrix, right? Basically, I create for each row, a set of numbers that obeys a certain property, which is that I assume that no strains are more fit than the other. And so, the sum of the resource consumption rates must add to a constant. There's some trade-off. If you're better at consuming something, you're worse at consuming something else. And so for each community, for each sort of set of strains, I get one matrix from this randomization procedure. And now I can instantiate the environment with a whole bunch of resources. What I do is I assume that all of the resources are available at roughly the same abundance to start with. And I run these dynamics to completion.
The resources are part of the differential equation.
Correct. Yeah.
Principal things are determined, right? So
correct. Yeah. Yeah. So once you give me Ci alpha, and once you give me the initial condition.
Oh,
right.
That's what.
Right. So the initial conditions have to do with how much resources and how much strains are present in the beginning. And so I imagine that all of the resources are roughly, similar. And then I run these dynamics over time and get my steady state. So, and then I sort of transfer a small amount of them into a fresh medium again with the resources. This time not exactly the same. This time slightly different. Maybe instead of all of them being R, there may be R prime, which is R. Yeah. So this model actually has a Lyapunov function, it turns out, and so it's stable with respect to initial conditions, and it has a unique stable equilibrium. Yes, so every time the resources get consumed and the species have grown and consumed all of them, I take just a small fraction of them and transfer them to a fresh medium, which contains all of the resources again, but in slightly different fractions. That's correct. Yeah. So in terms of the differential equations, I now start them again. The initial conditions of N I get from the previous transfer. The initial conditions of R I sort of get by randomness. And the CI alphas all remain the same for the same community.
Sorry, so
Yes.
Basically, if I set this tunable distance to distance d to 0,
yeah,
all the rows corresponding to a given, or rather, all the rows corresponding to a given species would be the same, right?
That's correct.
So basically, then I get to hypothesis 2.
Except with the following addition, that in hypothesis 2, I'm also explicitly adding my root and noise.
Okay.
My birth.
So without that, in this case, I would expect the strain correlation to be maximal, right?
Yes, yes.
Now, if I actually go to the plot that you showed from the sort of results of your simulation of hypothesis 1, you have the distance on a log scale, right?
That's correct.
So in a way, this is going down very quickly, like already by 0.1.
That's correct.
So basically, like 90% of your range, you would say, is in the decoupled.
Yeah, that's correct. And in fact, this is actually, to me, not so surprising, because the same thing sort of happens in the data. Remember, the sort of fall off happens at about 100 or so mutations, which is only 0.01% of the genomes of these different individuals. So the fact that, this transition occurs quite quickly is actually something that I think happens both in the model as well as in reality.
But aren't there very few points left of 0.1?
No, yeah, that's what I was going to
that's a good point. So the issue is that all the points are sort of all sitting at basically 1, then they're sort of masked by the red line. And that's just sort of to reflect that when the distance becomes so small, then you really can't.
So so mathematically speaking, is this coupled phase stable? Like, can you do like how do I know that this is not just an artifact of the simulation, and is this coupled phase perturbatively stable? Can one show that mathematically?
That is a good question. Maybe we can talk about it after the talk.
Sure.
Yeah. Okay. There's another question. Are the bacteria transiting from exponential growth to stationarity between each transfer? Yes, they are. What is the state of affairs in the picture likely to be? Okay, this is an excellent question. So the state of affairs in the picture, so usually so I'm not a pitcher plant biologist, but what my collaborators tell me is that what often happens in these pictures is that there are sort of boom and bust cycles. There's sort of, rain that occurs, or, something else that happens that sort of roughly resets the system, and sort of, washes away some fluid in the pitcher, brings in new nutrients, et cetera. And, a lot of the time that these microbes are growing because they get an insect, and then they deplete the nutrients, and then they move on. So they actually think that the serial transfers of growing and stationarity and transferring is actually not a bad approximation to what is going on in reality. So in both cases, there are two broad categories of genes being selected for. It's quite possible, yes, one set of genes that help you grow really well, and one set of genes that help you survive the stationary phase really well. That's correct. Sorry. Okay, great. Okay, so all right, so let me go back. So I was saying, that was the first pattern. The second pattern that our model is able to recapitulate is the one that I showed you here, which is that basically in 76% of cases, strain correlations are higher than species correlations. Just to remind you, this is what the data looked like. And what we were concerned about was this number 76%. And sort of here's what we get from numerically simulating both of our different hypotheses. The one on the left corresponds to strain-specific interactions, strains being the unit of interaction, and hypothesis 2 represents species being the unit of interaction. And we see quite a contrasting pattern here. What we see here is only one of our hypotheses is consistent with the data, that where strains are actually distinct units of interaction. And so what I'm showing you here is just an example from one particular simulation where I use 10 different communities. And so in this example, in 78% of cases, we had strain correlations being higher than species correlations. I can build a distribution, and we find that actually the kinds of numbers that we observe are actually quite typical from these distributions. In contrast with that, in hypothesis 2, we see the exactly opposite trend. In this case, we see in 81% of cases, species-species correlations are higher than strain-strain correlations. And if you think of it from the context of the model, it might not be so surprising. The reason being that in this particular model, it's really species that are the unit of interaction. And so two species trajectories are really correlated with each other. What is happening to strains within a species? They're sort of dominated by these sort of number fluctuations and birth-death noise. And so when you start to look at correlations between two strains, what you're looking at is essentially the correlations between two trajectories with noise added to them, right? And so it's no surprise then that two trajectories with noise added to them have a lower correlation than two trajectories without any noise added to them. Okay, so the last thing that I want to explain about this is a nice geometric interpretation that actually explains why it might not just be this particular case in which strain-strain correlations are higher, and why indeed it might be a generic phenomenon across almost all microbial ecosystems. And the argument goes as follows. The geometric interpretation is that in this model, you can think of all strains as being points that lie on a space of resource consumption rates, on sort of a resource space. And so just for simplicity, I'm showing you a resource space which has 3 resources, but my argument will hold for, will sort of generalize for higher dimensions as well. And so this space is a simplex because we assume this thing called metabolic trade-offs. That is, if you're good at consuming one resource, that you're going to be necessarily bad at consuming something else. This is reasonable from a biological standpoint, and that's what results in this sort of simplex here, because the sums of all consumption rates add up to a constant. But again, if we can relax this assumption and still get roughly the same results. So each strain is a point in this simplex of resource consumption rates. That's strain A1. What about its partner strain A2? That's for that we go a distance d away in this space and pick a point at random on the circumference. That can become a possible strain A2. What about now strains from another species? For another species, we pick points at random. So the strain B1 can be a random point on this simplex. And again, we go distance d away, pick a random point on the circumference, and that's strain B2. So we have all of our strains. Now we need to talk about the species that these strains are sort of comprising. And it so turns out that one can show that the effective species, A effective and B effective of each of these strains, can be thought of as these sort of spiky points lying on the line joining between the two strains. You can sort of do the mathematics of this model to sort of show that that's true. And so the important thing here is not exactly where it lies in between these two strains, but that it lies somewhere on the line joining between them. And so now when you construct this example, you can see why, here strain-strain correlations are often so often higher. So what you should focus on here are these 2 points, strain A2 and strain B2, which you will see are often closer than the corresponding species points, the corresponding spiky points. And remember, a few slides ago, the model told us that the functional distance between two strains can serve as a proxy for their correlations. And so in this case, what we get is that these two strains from two different species, because they're closer in this space, they're likely to have a higher correlation than the corresponding species themselves. In fact, you can try this on a notebook or on the blackboard, trying to make various versions of this. What you'll find is almost always this is the case, but not always. In a few cases, we do see species-species correlations being higher, and that's also something that the model can explain. The way to see it is by means of this example. Here's strain A. This is sort of easier when the distance d between strains is quite large. And so in this case, we have strain A1 here on the top right, and a distance d away, some strain A2. the corresponding effective species is this sort of spiky green point. And again, we have strain B1 on the top left, and a distance d away, its partner strain B2. And again, the corresponding species is on the line joining between them. And so maybe now you can appreciate that in this particular case, the two corresponding species, because they're closer in this resource space, we'll have a higher species-species correlation than any of the corresponding strain-strain pairs from across species. And so in this way, you might also be able to see why this would be quite rare. And so this is sort of why we think that the model is kind of explaining that because the typical behavior if strains are distinct will be to see any two strains from two different species being closer. The genetic consequence of that would be this kind of pattern that we observed in our experiment. Yes, yes.
I'm not sure I really understand the model,
Okay.
the geometry
Okay.
picture. I mean, are you trying to say that if you average over the points on that circle and this average distance between 2 points on these two circles will on average be larger than the distance between those two crosses?
That's correct. Yeah.
And so that's a statement. I mean, because this is an atypical configuration, right? There could be things on the far side.
Correct. Yes.
So you're
Yes.
making a statement about the average.
That's correct. Yeah. Correct. That is, if you try to do this again and again and again and compute these distances, you will on average find that that the strain distances are lower than the species distances.
That sounds a little unintuitive because you would think that those two crosses, they are fixed, right? And they are
the crosses aren't fixed. The crosses are always going to be on the line joining between the 2 points of the same strain. So if this strain sort of goes here, the cross will also move here.
Oh, it's on the line joining. Okay.
Right.
Yeah, but it's not obvious to me that the average will be indeed.
That's right. Yeah.
Because if you do have that configuration of the kind that you said.
Yeah. Then you'll have the centers being closer. Yeah. Then you'll have the centers being closer than the crosses, and it's not the points on the circumference that will be closer. But the point is still that there are two strains that are closer to each other than the corresponding species themselves.
How do you show this? Just by doing it again and again.
That's correct.
You don't have a real understanding of why this happens.
A real understanding of why what happens?
There's no theorem here. You just many tens of times.
Right.
To observe a pattern, basically.
That's correct. I would say the main sort of theoretical object here is the fact that strains are some distance away rather than being at the same point.
Yes.
And the second is the fact that the effective species can be always thought to be on the line joining between those two strains. And these are both things that that are mathematically true rather than something that we compute.
So I'm a bit confused as to why is it justified to put the cross as the mean of the two circles that you have done? Because I would have thought that a representative point for the species would be an average over all the strains for a given species. Right. And then that's not guaranteed to lie on the line, right? Because there could be in principle other strains which lie somewhere else in that circle.
Correct. No, you're absolutely right. So it so turned out that in this particular, like in all of our communities, we didn't have cases where more than two strains were present in a species. And so, this particular example was built for two strains just because two strains are what we saw. But you're right, if there were three strains within a species, then this theory would have to be modified for the spiky points to lie somewhere on the curve, within the convex hull of each of these points. That's correct.
And then, I mean, if that were to be the case, then I would imagine that, then I can understand that, of course, I can go and cherry pick the two strains that happen to be the closest. And that would give me the maximal correlations between the maximal strain correlations, which you were picking out in the first place in your previous plots. So would that be sort of the justification for why this geometric model?
Yeah, no, you're right. I mean, on one hand, it might seem like cherry picking, but on the other hand, I think it's a reflection of what's actually happening.
Okay.
That is the case that, most of the times within species, what you see are two strains that are functionally different. And it's often strains from two different, what we currently call two different species, actually are wrongly clubbed separately. They should be clubbed together because, when you try and construct them randomly, what you often see is that strains from things that you are calling two different species actually ought to be coupled together.
Right.
Okay, so I'm going to do I have 5 minutes to just end? Okay, so I'll take my last 5 minutes to sort of talk.
Sorry, just one question.
Yeah.
I'm still confused. So when you say typical behavior, so if that is the typical behavior, this is the consequence. That's what you're saying, right?
Correct, yeah. The typical behavior is that a pair of strains from two different species will be closer than the corresponding species themselves.
But I mean, how do you do it? I mean, how do you get it? I mean, is this what you're? I mean, how do you get this typical behavior? Do you, by your observation, or I mean,
right? So
is it? I mean, you're doing some time dynamics, or by experiment you're seeing this? I mean, what exactly is this?
Good point. So here, of course, I only showed you two examples because examples are what I can show you. But by typical behavior, what I really mean is by going through this procedure again and again of picking points, of going a distance d away, picking points randomly, and so on and so forth. The typical behavior that you find is that two strains from two different species will be closer. So,
I see. So you pick up 2 points, then draw a circle around it, and then randomly choose a point on the circle, measure the distance.
That's correct.
So you do some analysis, and then you see that. I see. Thanks.
At least to me, if you intended this to make it obvious that the strains will have a higher correlation, I'm not seeing that because you said you picked those 2 points on the sphere at random, and there could have, I mean, if they were on the far side, both of them, then the distance between the effective species would be closer than the distance between the strains, right? So
it would be closer compared to the two strains that you put on the far side.
Yes, but it would be, you're saying between the original strains.
That's correct.
So there would be a strain.
That is, you can always find a pair of strains within the species that are closer than the corresponding species themselves. Right. It doesn't have to be the point on the circumference.
Okay.
Hi,
Yes.
I have one question. So in one of the plots we show, it was actually pointed out that even when the distances are very small, your correlation still drops a lot, right?
Yes.
Right. So even when the strains are closely correlated in the species, you might still have larger distances so as to not see high strain correlation, right?
Even when you have large distances, or even when
even when you have strains which are closer than species, right? If you look at the averaged out distance, and you have distance between two strains, and you are arguing that the strains will be closer, then they might still not have higher correlation, right?
Right. So so here's a point of clarification, which is that when we're talking about the distance, we're talking about the distance between strains that belong to the same species. Right. That's what the distance d is, which you're asking for to be small and let it be small. Then when you're asking for all of these other correlations here, you're always measuring them between strains of different species.
Okay.
So you're never really looking at the d again.
Got it. Thanks.
Okay, great. Okay, so let me summarize and do a last couple of slides, which is basically, okay, so just to summarize, there were three main takeaways. The first was that if you start to look at dynamics within closely related strains, we find that, again, species doesn't seem to be the right unit of clumping different individuals in a community together. In fact, as soon as you go beyond 100 mutations apart, strains can become very different or decoupled in their dynamics. The second thing that I showed you was that the strength of dynamical correlations in these communities seem to be strongest at the strain level. rather than the species level, which is what was conventionally thought. And the third thing that I showed you was that both these kinds of patterns emerge from a consumer resource model where we must impose the condition that strains are distinct from each other rather than being the same. And so I just want to take my last couple of minutes to sort of loop back to what I talked about at the beginning and sort of mention the kinds of research that I'm excited about doing going forward. So like I said in the beginning, sort of the main thrust of the microbial sort of microbiome ecology in general is to solve what's called the structure function problem. That is, how to connect the various functions and the functional outputs of these communities with their structure, with sort of what is composed within them. And so we know from the past sort of few years that when we mean function, we mean the rate of some process. We mean either the rate of carbon dioxide production or nitrogen removal from waste and so on and so forth. What we don't know how to do well, and that sort of hinders us from being able to predict all of these functions, is to talk about at which level we should be coarse-graining these communities. And so I think that's what we're going to need going forward. What I showed you today, hopefully, was that the convention of grouping by species is perhaps not the right one, but a possible way forward might be to group things by dynamical similarity. But what about the case when we don't have access to dynamics? This sort of case that I described to you today was an experiment where sort of my collaborators went and sampled these communities and looked at them over a long period of time. But a lot of the times, the kinds of data that we have, which are abundantly available in public databases, are kinds where we have different snapshots of communities. And so we still want to be able to coarse-grain them because we want to be able to predict some functional output. And so how do we do that? My sort of main sort of hope and dream is that we ought to be able to do that with a two-pronged structure. One is by means of algorithms that are able to directly find coarse-grained groups in the data themselves. And in some sort of preliminary work that I'm doing, we're finding that sort of ideas from machine learning can help us find efficient low-dimensional embeddings of these systems that look very much like what I'm describing. And then the second is that we need theory to sort of help us build expectations for what kinds of patterns and what kinds of coarse-grainings we should expect in the first place. And I hope that I convince you that the kinds of consumer resource models that we build today might be a way forward to be able to do that. And so with that, the sort of three main things that I think that I'm excited to do going forward, and that I think this field is really in need for, is to first ask sort of what are the right coarse-grained variables for these microbiomes, and importantly, how can we learn them from the statistical structure of the data themselves. The second is to sort of ask, what are the fundamental physicochemical constraints on these variables? We know that a lot of these microbiomes are fundamentally constrained by the laws of thermodynamics, of kinetics, of metabolism, and currently the kinds of consumer resource models, for instance, the kind that I showed you today, don't respect any of these constraints. And so in some other work that I have going on, we are sort of started to incorporate these constraints in these kinds of models. And the third is finally to ask, how can we predict and control the collective outputs of these microbiomes using these variables and constraints? And so with that, I will end to the experimental people that worked on the work that I showed you today were Leonora Bittelstone, Gabriel Leventhal, and this work was done in Otto Cordero's group. And I thank you very much for your attention.
Read out the question.
Okay, so the first question goes, overall, are the observations in the lab or in the plant indicative more of drift or selection? I would say that they're more indicative of drift, and I'd say that for the following reason. The reason being that, well, okay, I take that back. I think that there are reflection of both of these things occurring. Drift, because we don't see a lot of adaptive dynamics in these communities. These communities grow for sort of hundreds of generations, and we see very, very few mutations accumulating in these strains and sort of creating functional differences. But selection also, because there are interactions that are there in this community that are influencing each of the strains or species. When one goes up, sort of the other responds to that. So I would say that it's sort of a combination of the two. Second question, for technological applications, for instance, bioremediation, does your work point to the importance of working with microbial communities comprising the right strains of several species rather than clones of the same species? This is an excellent question. So I think what my work is pointing to is that, right, so we do need to look at the right strains, but what we can do is zoom in to strains of the same species and break them up based on whether they perform similarly or not, and then coarse grain back again, sort of cluster back strains, even if they belong to very different species, if they are very similar in their dynamics or function. So what we ought to be doing is to be looking at these communities at the level of the genome, starting and inferring something about their function, and then coarse graining based on that. Does that answer your two questions?
Akshit, can I ask a question?
Yes, please.
This is Madan here from NCBS.
Hi.
Hi. So I was wondering, why didn't you use a sort of a population genetics model, such as a Fisher equation, and do a coarse graining on that, and that could then be coupled to to resource dynamics, which will inform its function, its fitness. So.
Yeah. Okay. No, that's a good question. I would say that in most cases, there's a sort of separation between population genetics models, which deal with evolutionary dynamics and don't really deal that much with functional similarity, and the sort of ecological models, which then don't think about the evolutionary dynamics and think more about the interactions that are happening within a community. And so in this case, I think we didn't have clear data telling us that there were strong evolutionary dynamics in these communities. And so that's why we opted not to use something like a Wright-Fisher model, which sort of describes things really well when you're just talking about sort of different variants that are roughly identical to each other and differ only in some fitness. We actually here wanted to ask, what are the determinants of that fitness in the first place?
No, because I thought it would resolve a question that Vijay asked about genetic distances, because then we could compare it directly to.
Yeah, no, okay, so I see. So what I can imagine doing is that we can take elements from a sort of population genetics model to do sort of an evolutionary version of these ecological models. This is something that people indeed do in the field, where you can sort of try to introduce mutations into the model by essentially mutating things like CI alpha. So what you can do is you can start off a model with just one strain and have it mutated CI alpha and sort of become a whole community of different strains. And that's certainly something we could have done. But I think that would be sort of asking questions about what are the time scales and sort of things that.
So the CI alpha will have its own dynamics.
Correct.
And probably that will give you dynamics of the CI alpha.
We just didn't know enough to constrain those mutational dynamics.
Questions, Fanta?
You model this problem in terms of, just the interaction of N and R. I mean, do you have any idea about what about the nonlinear terms, higher order terms? I mean, after all, you're modeling a system. Right. There has to be some understanding of why those terms are small, or.
Right.
I mean, that's one question. And then the other thing, you mentioned that there was a pattern recognition program that you were looking at.
Right.
And how does that compare with your model? I mean, the predictions.
How does that compare to the predictions?
Yes, I mean, we modeled it in some population dynamics equations.
Right.
On the other hand, this algorithm doesn't care about all that.
Absolutely. Yeah.
But there must be some matching of the results. So I just wanted to know that.
Yeah. No. That's an excellent question. So in fact, you're right that the algorithm will just find some low-dimensional embedding. It doesn't know at all where this comes from, and so that's that. So yeah, So that's really why I want to use theory to be able to understand when those kinds of embeddings are appropriate or not. What we don't know yet is a connection between when in ecological models do we expect structures that are like this, essentially. So for instance, one thing that you can do is start off with the CI alpha that I showed you today, not make it totally random and mixed, but have a sort of block structure to it, right? Have a perfect block structure where strains are just, there's one yellow group of strains that just consume one set of resources, one blue group and one red group. They're completely distinct from each other. And then certainly if I provide all of the resources that they use, then I'll see this kind of block structure. Now, what sort of happens as I start to sort of add noise to this matrix? These are the kinds of things that we don't know yet. And this is the kind of work that we would want to be able to do, so that both we can benchmark these algorithms to know when they're not giving us nonsense for nonsense sake, but also understand something about how this coarse-graining actually emerges in the ecological systems themselves. Does that answer your question? Sorry.
Yeah, I have a couple of questions. So usually when we do coarse-graining in some systems, we have some kind of phenomenological or macroscopic description in mind, and on the other hand, we have some microscopic description, and then we try to do some coarse-graining with the, keeping that macroscopic description in reference. So probably you have told us, but what are the kind of macroscopic or phenomenological phenomena or phenomenology that you want to understand finally as a long-term goal using this kind of coarse-graining? Probably you have explained, but can you please?
That's a really good question. So one of the phenomenologies would be the fact that, like I showed you before in my talk, each of these groups might actually represent groups of organisms that perform a certain function. So in the example that I gave you before, there might be that this blue group represents a group of organisms that break down a polymer in the environment, right? And so what we would then want to know is, as the level of the polymer changes in the environment, how does the level of these various species that change? And so one way of looking at the coarse-grain picture is to say, okay, it's the level of the group that changes, and I don't really care about predicting what the individual species are. And that's sort of the purpose. It's really what we want to be able to explain in the end is the rate at which this polymer will be degraded, the rate at which starch will be digested in your body. And so if I'm able to do that with a coarse-grain picture better, that's sort of what I'm interested in.
And the second question is related to your geometric interpretation.
Yeah.
If you can go to that slide. So and so this interpretation is said that if the distance is smaller, then the strain correlation is larger. Is because of the linear model that you have defined, and because of the way you are doing the experiment. Like every time you take something and then again reset the. Because otherwise, if you can choose a different initial condition for the different populations, and then you could get a completely different data point, which could have smaller distance can produce smaller strain correlation also, right?
Let me see if I understand. If I understand what you're saying correctly, you're saying if from like currently when I go from transfer to transfer, I take I use the initial conditions of the next transfer from the previous transfer before.
What I'm trying to say is that suppose I have infinite resources.
Yes.
So the resource is not evolving; it's always there. Yeah. So then also this connection will be there. That's my question.
If there are an infinite number of resources, yeah, I think so. I think that there's...
And is there an easy way to understand that?
No, I don't think he meant the infinite number. He meant the amount is infinite, like a bath. There's an infinite bath of resources.
If the concentration of resources are infinite. Yes, if the concentration of resources is very, very large, then essentially you don't even need to think about this as a community, right? Every organism is sort of growing exponentially by consuming everything for itself. And there's really nothing that they all
I don't need to think about.
Right.
My question is with this distance to correlation, this connection will still.
Yeah, I think it will. I think it will go away.
No, in some sense, I think the bath is the thing that is coupling the different communities, right? Because you're sucking stuff from the same bath.
Right. Like the reason that two strains are correlated in their dynamics is, for instance, they're competing for the same sets of resources, right? So if one of the strains consumes a lot of that resource, that
No,
then leaves
I understand that if you have in the first model, if you have D smaller, that means their rate of consumption. So these points are basically Ci alpha. Right, specifying the Ci alpha, and you're
that's correct. Yeah, this is a space of Ci alpha.
So if they're very close to each other, so they have the similar rates of consuming this thing. So they have similar population abundance curve. And you are finally computing correlation between the abundance curve.
That's correct.
So that's why you'll get a high strain correlation.
Correct.
Right.
Yes.
So I'm saying, and this is because you are measuring this correlation in the steady state where you have more or less stable fluctuations.
Right.
So this is because the way you are doing the experiment, but I'm saying if you do the experiment in a different way, like you have infinite resource and you don't do anything, it just evolves. And it goes to the same, I think it will have the same, by the same explanation, same smaller distance will have the same larger strain correlation also.
Right, just because you're consuming resources, you're repeating those resources in a similar way. Right.
Sorry to ask so many questions, but
no,
just very curious. You know, you model this system in terms of continuous time.
That's correct.
But perhaps more correct thing to do might be to really do it in terms of discrete time, like the logistic map, for example. Have you thought about that type of thing? Because, okay, that's easier to do,
right.
Perhaps.
No, yeah. So, strictly speaking, what we did was sort of a hybrid of continuous and discrete. And let me remind you why that is. What we're doing is that we are sort of mimicking what was done in the experiment, which is that there are discrete growth dilution cycles, right? So within each cycle, the dynamics are continuous, but between each transfer, the dynamics are discrete. So you could just sort of have all of the dynamics of these species where you don't care about the internal dynamics of each transfer. You just like a map, ask for the abundance at transfer, at the beginning of transfer 1, at the beginning of transfer 2, at the beginning of transfer 3, and so on and so forth. And that's indeed what we did.
I see. Okay. All right. Thanks.
I have a question about the biology, in a sense, underlying this, and when you say that there are 100 or 1,000 species in any such system, and then you're tracking you're grouping them by their functional similarity. Why biologically why is there a diversity of organisms with the same functional capacity or functionality?
That's an excellent question. Part of that question I will describe in gory detail in my chalk talk tomorrow, but I'll tell you the sort of short answer for it. And the short answer really is that, this point about saying functional similarity is, I slipped something under the rug here, which is that there's no one function that each of these microbes perform. But there's like hundreds of different axes along which you can measure function. And when we say functional similarity, we mean the similarity of some notion of what is the important function in that community. There are, of course, hundreds of other functions that these things are performing. For instance, they are constantly being attacked by antibiotics, or, other predators and so on and so forth. And so the notion is that there's diversity despite being functionally similar in these one or two things, because there's all these other nearly neutral traits that are nevertheless contributing to you staying in the same community.
And you are choosing this you're choosing to look along a particular axis because maybe you want to understand, say, the role in efficient digestion or efficient recycling or whatever, so there you can clump up
that
many
sort
of
of breaks the symmetry of which functions might be more privileged than other functions.
And even within a given function, what you showed is a correlation, but they might be actually playing different roles within that function, and so they could be kind of
sub-differentiation?
For the same, I mean, maybe there are two aspects to that function, but both of them are, both these bacteria are needed to.
In fact, again, in this polymer example that I was describing, often the way that microbes break a really long polymer is that, you know, some of them sort of chop up sort of one kind of bond and some of them chop up another kind of bond. And so that's the kind of sub-differentiation that might occur.
Okay.
Yeah. Hello. Yeah. So maybe you said this and I missed it. But what is the role the population size plays in this thing? Like, let's say relative to resources, let's say I keep a fixed amount of resources, and in this growth dilution cycle, I kind of, you know, the population that I take and go to the next cycle, if I change it, do these correlations that you see, do they change? Is there like some kind of a phase transition that there are correlations for a certain size or something? Or
so you're asking if I change the total population size, which I'll do by changing the total amount of resources that are present in the medium.
Right? So it's relative to that.
Right. Will there be a transition? That's an interesting question. I believe not, just because all of the terms here are highly linear, but in certain more complicated examples, and we can talk about this separately, there are actually cases where that happens. And that is because you can sometimes have growth that is cooperative. These are something that ecologists call alley effects, where basically the growth is itself a non-monotonic function of the population size. And so certainly in those cases, you can generate very different outcomes. And in fact, you get very often bistabilities.
Yeah, I was also asking whether these correlations between the strains, is it something that builds up, is there a certain population which is necessary? Like you need, or even at the number of species, we need some number of species for this to build up?
Oh yeah, that is an excellent question. So I won't fully describe it here, but it turns out that this whole thing that I described in the model actually only works when you have a sufficiently large number of species. And the reason for it is quite spooky. And the reason really is, like I was saying to Rajesh's question before, you need a sufficiently large number of species to survive. And even in this model, if you do this model for just 2 species or just 2 strains, everything collapses because you just can't get that small number of species to coexist with each other. And so quite paradoxically in these models, coexistence of large numbers of species is actually easier than coexistence of small numbers of species. And it's just because when you just have a very small number of species, they're all really competing for these nutrients, and they even small differences in consuming a certain nutrient can lead to vastly exponential differences in growth.
Okay. One more question.
Sure.
So which is like, if you take, like let us say it has reached some stable equilibrium, there's some correlations built up. If I introduce a new strain in, let's say, like a few number of species, like, can it destabilize this correlations? Like
if I introduce a new strain?
Or,
it can definitely affect the correlations. The reason being that, if I introduce something that's in the middle here, then of course it'll, that will start depleting resources more similarly to this. And so that might change some of the correlations. But again, in the high end limit where we are operating, it won't matter so much.
Unless you introduce, like a fraction of n number of new strains.
Yes, correct.
Okay.
Yes.
Okay.
Other questions? People online? Sanjay, Vidya, Madan and Vidya. Sanjay is not here, Madan is not here. Any other questions, comments? Okay, so if not, let's thank Akshit.
Thank you.
So it's nice to hear people actually clapping rather than Zoom clapping things coming up. Anyway, so Akshit will deliver another talk tomorrow at 3:30, same place. We'll catch up then. Thanks.